Our project is a subject pulmonary embolism identification. We did our project in Zebra Medical. Our wide data mentor was Tal Heletz, and our Zebra mentor was Mark Robbins. Zebra Medical uses deep learning to detect a variety of clinical positive findings in X-ray, CT, and mammography. So pulmonary embolism is a blockage of artery in the lung caused by a blood clot. It results in chest pain, shortness of breath, and even death. So fast diagnosis is very important, and usually it's done by CT. Every CT scan consists of hundreds of snapshots taken along a specific axis. We call them slices. And here we see an example of two slices along the axial axis. And you can see in white, this is a healthy artery, and the one thing in gray is the blood clot, it's a pulmonary embolism. So the business problem. Does a specific CT scan shows PE? What kind of PE? And which specific slices contain PE? This is a very time-consuming problem for a radiologist, which has to go over hundreds of slices, and the PE only is found in very few of them, and it's also very error-prone. As we can see here, sometimes it's very hard to detect the PE. It's very small and unnoticeable. So the RSNA issued a competition in Kaggle, a multi-label classification of PE in CT scans. And our base task was to reproduce the result of the Kaggle winning solution. The advanced task was to improve these results. And we have a super advanced task that we'll talk about later. So the data was actually CT scans that we call them studies. We have like 8,000 CT studies and almost 2 million CT slices. And each study or each scan has on average 250 slices. And we have like 10 labels. And all of them categorical or binary labels. And one label, like for the slice, was either is a PE on the slice. And for the study level, does this scan contains PE? Is the scan is indeterminate? Maybe there was some kind of problem during the scan. And we have also 7 other study labels regarding the location and the severity of the PE. And it was really challenging data as it was really imbalanced. Only 5% of the slices contained PE. Only 30% of the studies, the scans, contain PE. And also in each positive study has on average 18% positive slices. And you can see also the other study labels are very rare, like maximum 25% and the minimum is like 3%.
Okay, hi. So we talked about the data. Now let's talk about the competition evaluation method. So this is a multitask learning problem, and our KPI is to minimize the weighted BCE loss. Now we have different weighting methods for the slice loss, which takes into account the slice label weight and the proportion of positive slices in the study, and the study loss, which takes into account only the study label weights. And overall, we sum over all of these losses, and this is what we optimize for. So our first step is to reproduce the winning solution in the competition. Now, the winning solution has a pre-processing stage and then a 2-stage training strategy. So we have a CNN, which gets the images and predicts the slice label, and we use it to extract a features vector for each slice. And then we have an RNN, which gets a features matrix for each study, and it predicts both the study and the slice levels. In the pre-processing stage, we transform the pixels values, and we use a technique called windowing to highlight in the images the relevant structures related with PE. We trained a efficient net with a detection head to detect the relevant area in the images, the lung's bounding box, and crop it. Our 3 input channels for the CNN were the current slice and its 2 direct neighbors to mimic some kind of a volume. And we use these data augmentations. So for slice level modeling, we trained ResNext 50 with a squeeze and excitation block, pre-trained on ImageNet with BCE loss. The model was overfitting on our data, so we used early stopping, took the checkpoint with the highest validation AUC score, 0.96, which is the same as reported. And for study level modeling, we trained a bidirectional GRU. The study labels were predicted by concatenated attention and max pooling layers over the output sequence. And in terms of the input for the RNN, we adjusted each study to the fixed sequence length by resizing using interpolation or zero bedding. And we also computed the embedding difference between the current slice and its two direct neighbors and concatenated them. So this is Kagel's validation loss, and it's basically here to show that we've managed to reproduce the winning solution results. And now we want to connect it to our business problem. So we don't really care about Kagel's validation loss, but what we do care about is our model performance on the predictive task. So these are the AUC scores for the different study labels, and by Zebra's advice, we've decided to focus on predicting P on slice and on study. And in order to try and improve, we want to understand better what's behind these numbers. So by examining the distribution of P on slice AUC scores for different studies, we noticed that studies with relatively lower AUC scores have lower mean percentage of positive slices. And this is intuitive, right? Lower percentage of positive slices means less severe PE. PE, which is harder to identify. We also wanted to check if we can say something about the study by just looking at the maximal prediction for PE on slice. Is it enough for predicting PE on study? So we see there's a strong correlation between them. We sampled some false positive and false negative studies from this plot and examined their slices with Zebra's radiologist. So for the false positive, the radiologist said there's an RV-LV ratio much larger than 1, which is a major indicator for PE. And for the false negative, in one slice, she said it's a difficult case to identify, and in the other, she didn't notice PE, which may indicate there's some noise in the labels. So based on this analysis, we're trying to improve the current results. What eventually worked? Implementing focal loss, which assigns more weights to difficult samples, and also implemented a weighted sampler to upsample the PE positive class. This way, we've managed to improve the CNN and the RNN AUC scores for P on slice and on study, and as their ROC curves show, for the same false positive rate, we get a slightly higher recall. But there are some problems with this solution. By deconstructing Kagel's loss and examining each label loss separately, it seems like the RNN doesn't really learn anything. To dive into this, we examined the RNN performance. So in terms of predicting P on slice, comparing the CNN on the x-axis to the RNN on the y-axis, it seems like the RNN can separate these classes a bit better. But in terms of predicting P on study, by applying random forest, or even just a naive classifier, which takes the maximal P prediction on slice, we get comparable results. And this makes us question if the RNN really has an added value here. Okay, so until now we talked about how we reproduced and even improved the original model, and now we'll talk about the advanced goal we chose, applying self-supervision on our data. Why? Well, we had a lot of data, but usually this is not the case. It's difficult to get labeled medical data, and it's very expensive to annotate it. So often you need to be able to build a good model with much less data resources. So imagine a situation where we have much less labeled data, and much less means this is affecting the performance of our supervised approach. How can we improve it? To simulate such a situation, we trained Kagel's backbone on only 2% of our data. Why 2%? Because with 10% we still got pretty good results, which indicates we still had the same amount of variance. In order to try and improve the supervised approach, we tried two different semi-supervised models. The first one is SimClear, and it is based on contrastive learning. And the second one is fixed match, and it is based on knowledge distillation. So let's start with SimClear's architecture. In the pre-training stage, we use only the unlabeled data. For each unlabeled image, we apply 2 random data augmentations. We pass them through an encoder and then through a projection head to get representations. And finally, we maximize the similarity between them using this loss, where tau, the temperature, is a hyperparameter. In the fine-tuning stage, we use only the labeled data. We extract its embeddings from the pre-trained encoder, and then we use them to fine-tune a linear classifier on the downstream task. In our implementation, we used Kagel's backbone as an encoder. Our downstream task was predicting P on slice, and we had to adjust the optimization scheme and also find the optimal hyperparameters for our architecture. The major challenges we faced were choosing the right augmentations, so we did some kind of an ablation study, applying the transformations one by one to only one encoder branch, then choosing the ones which gave the best linear classifier performance. Scaling up really improves performance, so we downsampled the images and used distributed training to increase the batch size and train for a longer time, although we still couldn't reach the parameters, the epochs number, the batch size, the original paper talked about. And the pre-training process is sensitive to the temperature. So we did a log scale search and validated the correlation between the contrastive accuracy and the linear evaluation. But eventually we couldn't improve with this approach. So looking at the distribution of similarity values for the embeddings of P positive, P negative, and mixed positive-negative slices, we see they are all very close to each other. So we assume that the task of distinguishing one image from another based on some general features didn't allow the network to learn the specific meaningful representations for PE prediction, which is extremely small and extremely rare in the images. So SimClear's approach didn't match the nature of our data, but another approach did, and this is fixed match.
So alongside with SimClear, we tried another semi-supervised algorithm, a fixed match, and the core of this algorithm is a loss function. It consists of 2 parts. One part is a binary cross-entropy loss on the labeled data. This is like the old supervised loss as we know it. And the other part is a binary cross-entropy on unlabeled data. And for this, we need the labels. So we create our own labels. It's actually called pseudo-labels. For each unlabeled example, we have 2 versions, one weakly augmented and one strongly augmented. We pass the weakly augmented through the model, gets a softmax prediction, and if the maximum passes a certain threshold, then we've got a pseudo-label, and we compare it to the prediction the model gave on the strongly augmented version. And these 2 parts are computed simultaneously, so every batch consisted of both labeled and unlabeled data. And so fixed match has good results on CIFAR and STL, but hasn't been used on CT scans before. So we had to make a few changes. We used the Kaggle backbone and optimization scheme. We used different augmentations. We used smaller batch size, and we used the 2 thresholds, actually, minimum and maximum. We applied it on the same 2% of labeled data as SimClear. And first big challenge was that the batch size. The batch size is an important factor in all semi-supervised algorithm. And the original paper used 512 and up to 1600, and we used 16 because our GPU was quite limited. And also we had to use an equal amount of labeled and unlabeled data in every batch, and the paper used 5 or 7 times more unlabeled data. This way, as you can see, a lot of unlabeled, a lot of data in every epoch, and we could see very little. We tried to change the balance, we tried to lower the image size, but it just didn't work. Another big challenge we had was the pseudo-label. There's a very known problem with the pseudo-labels, that they become noisy labels. And here is a combination with unbalanced data. It was even a bigger problem because the noise is asymmetric. More PE images are misclassified, get the wrong labels and the other way around. So actually the network sees much more negative examples than in the original data, and it becomes more biased. Another thing, moreover, the noisy labels can mislead the loss function to reinforce irrelevant features. So we replace the hard pseudo labels of the paper with soft ones. So in case the labels are wrong, the incorrect loss will be smaller. So we tried all the imbalance classic as focal loss, weighted loss, upsampling. They all didn't work. We tried to make the network more robust and pre-train it on a slice location task. We noticed that there is an association between the slice relative location and PE. Like more PE is more prevalent in the middle slices. So, and this is like labels that we have in abundance. So we tried pre-training on this task, but it didn't work. And finally, we used the adaptive threshold. This is our own self-made variation on distribution alignment that appeared in the paper. But the idea behind both is the same. We want to encourage the network to produce pseudo labels in the same known distribution. So they have this distribution factor and they multiply the softmax width. But we tried it, but it was just too aggressive. And in the beginning of the epoch, the model is still unreliable, the distribution is still unreliable. We don't want to encourage too much PE. So we made 3 changes. We added this alpha, this data progress factor. We created this eta, and we changed the threshold. We adapted the threshold to change according both the data and the distribution. And it worked. We got a very nice improvement, AUC of 89.84 instead of the baseline of 87.35. You can see the improvement also in the ROC curve. Later we wanted to check what was the contribution of this adaptive threshold, what was the added value. So we trained with and without adaptive threshold. We could see blue is with and red is without. And we can see the impact on the number of labels, of the pseudo labels, and the loss, the unlabeled loss is lower. But on the other hand, we know that this is a consistency loss, and we can get very low loss also with noisy labels. And we also see here that we have less PE labels. So we tested directly the pseudo labels on a separate validation test, and we can see that with adaptive threshold, we get much better recall. That means that actually not only we get like more pseudo labels, they're also better. And without adaptive threshold, we get less PE labels, but actually we get more PE labels, sorry, but they're like more noisy, it's like more noise. So, summary, we had 2 basic goals. We accomplished them all. We reproduced the Kaggle winner results on a multi-label classification task of highly unbalanced medical data using a complex 3-fold architecture. We had this lung localization and feature detection and then classification by RNN. We managed to improve these results by up to 1.5%. We added an extra advanced goal. We implemented 2 state-of-the-art semi-supervised algorithm on 2% of the data and succeeded in getting 3% improvement compared to the supervised baseline. And most important goal, we really learned a lot. And huge thank you for Tal. I'm so excited because she's really #1 mentor. And huge thanks to, also thank you to Zebra and Heletz.